next generation technology platforms, starting with the structure of the DNA. Watson and Crick, they deciphered the structure of DNA using Rosalind Franklin's X-ray diffraction of bones. And in 1953, the double helix was discovered. But it took almost 24 years for Sanger to come up with a technology which is very popularly known as Sanger sequencing, and for the last 50 years has been the gold standard in sequencing DNA. So in 1977, that technology was introduced to us, and subsequently there were two technology platforms. One was polymerase chain reaction, and the other was automation of this Sanger sequencing, which allowed the grand human genome project to happen, which started in the 1990s till the first draft, incomplete draft of human genome was released to the public through a government-funded project. And this draft in 2001 called the Human Genome Project had some gaps, which took scientists 20 years done within the year to actually fill those gaps only recently. And now we have the complete 3.2 billion sequence in place of healthy individuals, so to say healthy. Now, what happened between that era? All this human genome sequencing happened using Sanger platforms, automated Sanger platform. It was in 2004 when the first platform of massive parallel sequencing was launched by 454 pyrosequencing. Roche was the one who launched that in 2004. And ever since, the technology is evolving so rapidly that even it is hard for us to keep updated. And what you see here, and since I'll be talking about oncology, the first whole exome sequencing of cancer was published in 2008, and that was in acute myeloid leukemia, and that's the area where we work on, liquid cancers, blood cancers. That was in 2008, and ever since, there are like 70 whole cancer genomes, exomes, so much data, bank has been developed, the TCGA pilot project has been launched, so massive data is available, but mostly from Western countries like Europe and America, Japan too, and so the field has been evolving ever since, and this technological revolution has brought us today to what my second half of the talk will be about, precision medicine. Now, so this is the overview of my talk. Like I said, in the first half, we will go through the principles of Sanger sequencing and overview of next generation sequencing, the difference between the two platforms, applications, and applications is where I will share with you a couple of panels that we're using in the lab and how we are helping in patient care using these approved targeted therapies, deriving information from these from next generation sequencing platforms. What you see here on the right is Fred Sanger, who invented Sanger sequencing, and he won the second Nobel Prize in 1980 for the determination of base sequences and nucleic acids. Now, what is Sanger sequencing all about? The film that you see in his hand is an X-ray film, autoradiograph, which even I, as a PhD student here, has used till 2003. In fact, this technology was introduced in 1997, but for the next almost 25 years, we were still using this in the old format until it was automated. So, what I will be showing here is the automated version of Sanger sequencing, and I will also tell you slightly the difference between the two. Now, how do you sequence a fragment of DNA? What you see on the left is a sequence of DNA. So in a tube, if you have a template, like say genomic DNA, isolated from cells or from a source, could be blood, could be bone marrow, could be fluids. Now that DNA, if you mix with a primer, which is called an oligo, and give it a DNA polymerase, that's how naturally this replication happens. But in vitro, if you take these components and you put it in a tube, the DNA polymerase, which is recombinant, now here, will and also give it a mixture of the bases, the D and TP, the deoxynucleotides. This will facilitate the, so if you denature the DNA, the two strands are separated. The oligo is going to bind to one of the strands, polymerase will keep adding the nucleotides in the linear fashion and complementary to the bases that you see. Now, to denature the two strands, we have the ABCD in a certain order. We need to now know the sequence of this, the DNA. A fragment of DNA, like I said, it's no technology is able to do the entire 3 billion. We have to use targeted fragments to sequence like certain regions, small regions. And this polymerase is going to add the complementary nucleotide. So if this is the strand, the denatured strand at 95 degrees, the TD is now in a single linear strand where the complementary base is A, which is here. So the DNA polymerase is going to add the complementary bases A, T, C, C, A, T, T. So complementary to the bases present in a single strand. Similarly on the other side. So this is how millions of copies are made in a PCR reaction, which came later on. But this is cycle sequencing. Now, what did Sanger do to create the sequencing reaction? He designed chemically modified dideoxy bases. Now, how are they different from DNT ones? You see there is a hydroxyl group. So as each base is added onto this primer, the oligoprimer, the hydroxyl is free for the next base to come in and attach covalently. But if you block that hydroxyl, the next base will not be added. So this is called chain termination. Now in a mixture in the tube, if low concentration of dideoxy entities are added to the tube, what's going to happen? You will generate a mixture of fragments which are terminated at the corresponding nucleotides, and this mixture, the length of these fragments will differ only by single nucleotide. And these can be easily resolved on a gel electrophoresis or now in automated capillary electrophoresis, and that can then be read. I'll show you the next slide. So this fragment, this is what is generated in the tube. These are fragments differing in one nucleotide, and they're fluorescently labeled. So the laser beam in the automated equipment is going to excite the fluorochrome and then emit a light which will correspond to that nucleotide. Alright, so here, this is the API sequence we have in the lab, 35 on here, 35. You see our staff, Pramod, is operating the machine. He's doing other applications other than sequencing also, which is fragment analysis. I think I had presented in the last and we have a mixture for expert in analysis. So this is one challenging sequence.
उसी के लैब के हैं सारे
He doesn't
मैक्सिमम जो प्रेजेंटर हैं तो वो है IRCH के हैं
where it's a single codon gene, and the mutation is not a hotspot mutation. It is randomly scattered all over the gene sequence, and sometimes it is a challenge, and I sometimes don't trust him when giving the data. Where is the mutation is like, and sometimes for him also it becomes challenging to identify those mutations, but he is pretty well versed, and I can blindly trust him when he gives me this information. So this is a software for analysis. So in this machine, that sequencing reaction I showed you is loaded, and then the capillary, this is capillary sequencing, 38 capillaries, which, and you use 96 cell plate format, and the capillary, the sample is injected into the capillaries, they migrate based on size, smaller fragments will run faster, but what you see is the electrophotogram, it is the inverted, so the smaller fragments are here and the larger fragments are here. Each peak in the electrophotogram represents a nucleotide, and this is how you read the and if there is a mismatch, which I will show you later.
हमारे डिपार्टमेंट से 2-3 लोग आए
Where if it is a mismatch, double peaks. Or a change of that.
लंच टाइम तक आ जाएंगे सब
पता है कहाँ पर रह गए आना चाहिए थे सोच में तब लोग आ गए थे प्रोग्राम
लंच टाइम तक आएंगे या फिर 4 बजे तक आएंगे
कल तेरा प्रोग्राम में तो ये हम लोग तो मॉर्निंग में आ गए थे डायरेक्टर सब आए थे नहीं मैं तो अभी आया हाँ वो तो देखा
800 bases of a fragment, and it has very high accuracy, but the major limitation of by Sanger. And then we depend on PCR technology, and that's where now I will show you how next generation sequencing has higher sensitivity to detect these changes. Now, although it does have long reads, but if you remember from the pandemic, most of the, so Illumina designed, and a lot of other companies also designed NGS-based panels to cover the
अबे चाय पिला रहा है ना, हमें ऐसे ही बैठे हैं
variants of
आगे
SARS-CoV-2.
आगे वीआईपी लोग
And
बैठे
why is
हुए
that?
हैं
Because the
बड़ा
genome is
VIP
30
बैठा है
kilobases, it's humongous. So if you do a standard sequencing, so here, one tube means one reaction. But to cover a genome of 30 kilobases, you will require, like, I didn't do the
सर्टिफिकेट
math,
तो
but
भिजवाएंगे
there'll
ना
be
बता
like
रहे हैं
multiple tubes of reaction that
दे
then
रहे
to
हैं
अभी कहाँ
reconstruct
जा
the
रहे हैं
sequence of 30
बाहर में है ना
genes,
सर्टिफिकेट
identify the
सर्टिफिकेट
variant.
कब दे रहे हैं
Instead,
अभी तो हमको
NGS
आज तो नहीं
panel,
दिए
it does massive parallel
हाँ तो बाद में
sequencing
लेंगे,
and
ले लेना
you
जाके
guys
अभी
want one
ले
single
रहे हैं
reaction.
ले लेना
That's where we
ले देंगे
So this is how the technology improved from, like I showed you the timeline, from gel-based
तुमने लिया
capillary sequencing, all standard sequencing, from the first generation sequencing, we moved to the
तो
massive
बैग पे
parallel
आया वहाँ
sequencing,
पे बैग तो ठीक
where
है
initially started off with
से
several platforms,
ठीक
which are in the next slide, and but they all they do short read sequencing. Now, in the last 5 years, we have newer technologies for long reads. These short reads refer to like less than 600 base pair, which is generally 300 to 400 base pairs what can be covered. But in the long reads that are now PacBio, as I'll show you in the next slide,
कुछ काम
these are
है
the technology
दो काम तीन
platforms, starting from Sanger, how it has moved the
सबके
first,
लाद लिए
कई लोगों का लिए
ये कह रहा
next
सबके लाद
generation
लिए
sequencing platform, which is the Roche biosequencing
आरपीसी वाले
platform,
हैं
then the AII
आरपीसी की
platform,
नहीं
the
है
ion semiconductor platform,
सर
Illumina
कैंसर वाले हैं
and now
ISCH
the
के
single
ये
molecule
ISCH
ISCH के
sequencing,
हैं
PacBio
आरपीसी
and
की सिर्फ वही एक
Nanopore,
मैडम है
and then yeah, these they the next slide I'll show you. So here